Hello friends, today we are going to discuss one of the important topic, gene rearrangement with respect to immunoglobulin light chain rearrangement. That means VJ recombination. That means gene rearrangement with respect to immunoglobulin light chain rearrangement. That means VJ recombination. Now, what is gene rearrangement? With respect to chromosomal rearrangement, that means nothing but a mutation that changes the structure of the DNA. Say, in the way of duplication, deletion, inversion, translocation. There are different classes of gene rearrangements. With respect to our today's topic, that means immunoglobulin light chain rearrangement, it is somatic recombination. What is somatic recombination? It is alteration of DNA in a somatic cell. Generally, it refers to large scale alterations like chromosomal translocations or deletions, which have detrimental or adversarial consequence sometimes, because after all these are all mutations, and the particular kind of gene rearrangement involving in immunoglobulin light chain is transpositions. It's like gene transposition of genes we have discussed in our previous classes. What exactly transposition of genes? They are jumping genes. A one particular fragment of DNA will cut down and again reinsert into somewhere, that means in somewhere in the genome. It's transposable. That means transposable elements, and they're called jumping genes. Say lines and signs we have discussed at length. Similar to that, the same concept here. Here, a particular fragment of DNA will rearrange with other to form, that means to assemble a light chain. What exactly I meant to say? We all knew that a typical immunoglobulin, that means antibody, what we call B cell receptor, contains two light chains and two heavy chains. And each chain contains two portions, one variable portion and one constant region. The variable portions of, that means light and heavy chains, in combination, it forms what we call paratope. It is the binding site for antigen. Epitope-paratope interaction. That is a basic mechanism for how the antibodies fight against the antigen. That is how we defend. That means our body, our immune system defend against the foreign invaders like bacterial pathogens. Okay, it's all about the immune system, but I am discussing about the basic structure. It contains two heavy and two light chains with variable and constant regions. Here, focus on the variable regions. Why? And what is the reason for this variability? The variations in the light chain and heavy chain is a crux point for diversification of antibodies. Our human body produces some around 10 billion kind of antibodies, such a vast number, 10 billion antibodies, which are essential in fighting against a variety of epitopes, because we may not know what kind of epitope does a particular antigen, say in the way of bacteria or virus or some other pathogen may have. So to fight against the same, should have a specific paratope, what we call specificity between antigen and antibody, that is essential in fighting against the foreign invaders. And this essentiality is bringing down by variable end of the what we call antibody, that means B cell receptor, for instance. And the mechanism for this variability is gene rearrangement. Gene rearrangement in the respect to DNA fragments, what we call in respect to or with respect to different or concerned genes of light chains, is a basic crux point for diversity of antibodies. So 10 billion antibodies. What I mean to say, the kind of, say we knew that, I would like to go with respect to a structure, it is a basic structure of antibody. It consists of two chains, one heavy chain, means two light chains and two heavy chains. And each chain consists of two portions, the variable part and the constant part. So it's a typical Y-shaped molecule. Say this is a variable part and this is a constant part. This is light chain and this is heavy chain. This is called VL, variable part portion of light chain, and this is CL, constant portion of light chain. This is VH, same variable heavy chain, and this is CH. This together is called FAB, fragment antigen binding site, and this is called FC, fragment crystallization. Say this is plasma membrane, that means B cell plasma membrane. That means the FC part binds as a receptor on the surface of B cell. So it's about the basic structure. We are just discussing about the light chain. Today we're going to discuss about light chain. The light chain. That means the variable and constant part. We knew that heavy chains are different kinds. Like alpha heavy chain, gamma, delta, epsilon, muon. There are 5 kinds of heavy chains. Similar to the light chains are 2 types, kappa and lambda. Okay, and the differentiation of different antibodies is based on the heavy chain, the kind of heavy chain. And so it's about basics, okay? And this is called particularly the epitope, to which the antigen will rest and bind here. Okay, through the respective epitope, what's called epitope-paratope interaction. But the thing is, the kind of epitope, according to the kind of epitope, the paratope has to be synthesized. This is very important. That means the variable portion of the light chain, heavy chain. That means 2 heavy chains and light chain is essential in interacting with the epitope of antigen, without which it is impossible to weed out the fault invaders. And what is the secret for that? I told already, diversification of antibodies at the rate of 10 billion antibodies we have. 10 billion antibodies we have. What is the reason? This because of gene rearrangement. This is very important point. With respect to light chain, we'll discuss how the issue ends. For example, say the light chain it is not one particular polypeptide. It is an assembled form. It has to be assembled from 3 different regions. What we call variable region, the joining region, and the constant region. That means in a DNA there are 3 distinct regions or segments. Say variable light chain segment, joining segment, and the constant segment. And these 3 segments has to brought together, what we call gene rearrangement, during B cell proliferation. This is very important. What exactly gene rearrangement means? That means VJ, I told you, variable and joining. Here gene rearrangement is particularly reference to VJ region. The constant region rearrangement takes place at mRNA level. Actually, with respect to immunoglobulin, G rearrangement takes place at 2 levels. At DNA level and at RNA level. At DNA level it's called VJ joining, whereas RNA level is called C region splicing with V region. Actually VJ, it forms variable portion which joins at RNA level through splicing. We have discussed at length during transcription. Splicing, that in a spliceosome we have to break down the introns, so the exons has to join through spliceosome, what we call small nuclear SNRPs. They involve, and that what we call complex of SNRPs, they involve and participate in splicing. And through splicing, the C region has to join with the V region, that ultimately leads to translation, and that's how the V, that means the light chain of the immunoglobulin, get assembled. That means the recombination of the VJ region, then the splicing with the C region is the mechanism of how we expect different varieties of light chains. That is like, for example, how it with particular example. For example, I told you light chain is synthesized from three different regions in DNA: variable gene, adjoining, and its constant. In general, in human DNA, there are 500 variable portions, whereas 6 joining portions and 20 constant portions. That means 500 segments of variable genes with respect to light chain I am saying, and 6 segments of joining and 20 segments of constant. This is very important. 500 for variable, 6 for joining and 20 for constant. It leads to different combination, what we call innumerable combinations. Innumerable combinations between among these three, that means gene rearrangement through the innumerable combinations through 500 variable, J joining, 6 joining and 20 constant is a crux point for production of variety of antibodies to fight against a vast diversity of antigens. That's how we defend our system. That means our body. That means through our immune system. That means gene rearrangement is actually involved different combinations. What I told you, different parts have had to brought together, like jumping, like transpositions. That's how the issue is. That means we have 500 variables joining 6 and 20C. Okay. Actually, the VJ rearrangement takes place at DNA level, but the constant region through splicing at RNA level it takes place. Ultimately leads to assemble of B cell receptor, what we call antibody. This is the basic mechanism and basic things of VJ recombination or means immunoglobulin gene rearrangement. With respect to specific example that I will discuss. Okay. I told you already, for better understanding, for illustration, we'll take one particular example. I told you already there are 500, V region, there are 500 segments of V. Say these are all for illustration, okay? These are all different 500 variable segments. And there are some 6 segments of joining. Say J 6 segments. Okay. These are at different positions. Okay. It means there are different positions in DNA and they have to brought. That means in the original DNA there are different positions. Later onwards, through gene rearrangement, the variable part has to brought to the joining and the constant part. That is what exactly called gene rearrangement, and it takes place during B cell proliferation. The primary lymphoid organs. What exactly it is. Say this is the final part, constant part. Okay, this is constant, it has of 20. Okay, this is the original DNA. Then it will be subjected to gene rearrangement. I told you already, the variable, say we'll take particular example, 100th variable has to be rearranged, has to be brought closer to a specific joining, say joining third joining region, third joining segment, with the ended up the 10th constant segment. Say, this is how the issue ends. After rejoining, we got this. We got this after rejoining, what we call rearranged DNA. Okay. Then it will substitute, primary transcript. The same we will get. This primary transcript. Then, I told you already, post-transcriptional modifications, particularly mRNA splicing. The C region has to brought, that means the VJ region, which has been brought together, has to be brought close to, that means C region through splicing. No, this is what exactly I meant to say is the 100th part. Say this is V100 and this is J3 and this is C10. Okay. With respect to one particular segment I'm discussing for better understanding, then we'll get the final form after post-transcriptional modification. So what we call PTM. This is one, and finally this is the end product. This is the 100th part. That means V. This is the third joining, the 10th segment that leads to synthesis of light chain. What we call assemble. The assembled part. The assembled part leads to synthesis of a specific polypeptide. Say this is a V variable and this is a J and this is a C. That means light chain. This is how the issues. Actually VJ they together form variable part and constant part is the second domain. That means initially there's say for example I have told 500 segments in variable segment, 500 variable segment, 6 joining segments and 20 constant segments in original DNA. But after gene rearrangement, after they all reorganize, the 100th one specific example, 100th variable brought down close to the joining third and C10. Then primary transcription, we knew that. Then post-transcriptional modifications, what we go through splicing, the constant region brought together that ultimately synthesize a light chain. Is how the issue ends. I told you already. The innumerable combinations of among these 3 DNA segments, variable, constant, and joining is the basic mechanism of production of a diversity of light chain. That means different light chains. This is with respect to immunoglobulin light chain, what we call VJ joining. And how about heavy chain? It's on the same mechanism, but a different, but a like bit different. What we call V delta J joining. Because here D means diversity, diversity is a one more gene fragment. That means here in case of light chain there are 3 gene segments, variable, the joining and the constant. Whereas in case of heavy chain, variable, diversity, the second element, then joining and constant. That means 4 gene segments will involve in heavy chain, that what we call VDJ recombination with respect to heavy chain. Whereas VJ recombination with respect to light chain. These are the 2 things. But the concept is same. But maybe the basic mechanism will be same. That means first they have to brought together what we call VJ, then the constant. That means the splicing, one at the DNA level, and the RNA level. Ultimately form heavy chain. I told you already there are 5 heavy chains. The IgG, gamma, which is a monomer, delta, IgD, Ig epsilon, IgM, muta. That means 5 kinds of antibodies and 5 kind of heavy chains, and it is a basic mechanism for classification of 5 classes of immunoglobulins. Whereas in light chain there are 2, kappa and lambda. So rearrangement amongst these 3, that means among these DNA segment, that means light chain and the heavy chain of respect to say the variable light, that means variable joining constant with respect to light chain, variable diversity joining constant with respect to heavy chain is the basic mechanism for production a vast number of antibodies, say 10 billion antibodies for what we call specific antigen antibody interaction, that ultimately we can fight against these things. That means, so for all these things the basic mechanism is gene rearrangement, and the kind of the specific rearrangement is here, it is somatic combination through transpositions. Okay, this is how the issue ends. And in some other class you can discuss some other topic. Okay. In addition, the main objective of my presentation is to cater to the needs of needy and poor students, and you have to rely upon my standard book and a basic book, so that you can consolidate your knowledge with my tutors or lectures. Thanks for watching.